All right, at this point, we've talked about translocation of proteins and transport to the mitochondria, the chloroplast, the nucleus, how it's translocated into the ER. We've even talked a bit about vesicle transport and the retrieval pathway for ER resident proteins. We also know that from the Golgi, by default, things will be packaged in vesicles towards the plasma membrane. But what about things that are meant for the lysosome? So obviously to get to the lysosome, it should not go the default pathway, so there needs to be another tag or another address marker to get it to the lysosome. So that's what we're going to talk about here. So the things that need to get to the lysosome are all those ACEs. So the nucleases, proteases, glycosidases, lipases, phosphatases. These are things that break down very specific parts. And this is obviously very, very important because your body needs to break down old proteins, needs to break down old nucleotides. It needs to basically get rid of the junk and hopefully recycle them. So if you break down a protein, you can recycle the individual amino acids and use them to link together as new proteins are made and so forth. But if these aren't working, it's pretty much like what happens if you stop cleaning up your room. If you stop cleaning up your room, things are going to build up, and pretty soon you've got a big issue. And so if things build up in the cell, that causes either cell death or sometimes even disease. Things like Alzheimer's, for example, and a lot of other neurodegenerative diseases are caused by an abnormal aggregation of proteins that build up and basically create junk, and therefore the neurons stop working appropriately. So it's really important that these lysosomal enzymes get to where they're going so that your cells can get rid of its waste, essentially. So the way they get there is these ACEs, these enzymes, have an ER signal sequence. Then they move on to the Golgi, and after the appropriate folding and sugar modification, they will be tagged and then go to the endosome, which endosome then kind of becomes the lysosome. So the pH will steadily get lower as it's on its way. And the pH we know in the lysosome is much, much lower. Part of that's due to this pump that basically actively pumps protons in here, lowering the pH for this degradation organelle. All right, so the signal that sends these enzymes from the Golgi to the lysosome is actually a sugar residue, mannose 6-phosphate. So M6P, mannose 6-phosphate, is the signal that says this is a lysosomal enzyme, send it to the lysosome. Right? And so these are added by enzymes. So the lysosomal enzyme will have its glycosylation, N-link glycosylation, and it's going to have a terminal mannose residue here. And then GLACNAC phosphotransferase basically is an enzyme that's going to recognize this lysosomal enzyme, and it's going to add this GLACNAC, that's a different sugar, with a phosphate group onto the end of the mannose. And then the next enzyme, GLACNAC phosphoglycosidase, basically is going to take off this sugar, leaving the mannose with a phosphate residue on carbon number 6. Hence mannose 6-phosphate. Mannose is phosphorylated at carbon number 6. So we have two enzymes that are needed to do this. GLACNAC phosphotransferase and then GLACNAC phosphoglycosidase, and at the end of that the lysosomal enzyme will have its M6P, and that sends it to the lysosome. But here's the thing, these enzymes don't recognize a signal sequence. They don't recognize an amino acid sequence. They recognize what's called a signal patch. So what do you think the difference between a signal patch and a signal sequence might be? All right, well, a signal patch is a three-dimensional folded region of the protein that kind of has that signal. And it's not a sequence of amino acids that are one after another. It's this folded patch. And so the patch may actually be made up of amino acids that in the primary sequence are a long ways away from each other. So this might be a couple of amino acids at 12 and 13, and here we're at 140 and 141, and so forth, and near the end of the protein. So all of those come together in the fold and make that signal patch. So the next question is, why do you think the lysosomal enzymes require a signal patch rather than a signal sequence to basically signal that it needs the M6P to be added to it. So in other words, why would these enzymes recognize a signal patch instead of a signal sequence? Like, for example, we know that the ER signal sequence to get into the ER in the first place is a sequence of amino acids recognized by the signal recognition particle, SRP, right? But here the enzymes recognize this patch. Why might that be? Well, the reason for that is because by the time we get to this medial in almost the trans Golgi, this protein is nearly fully folded. So a signal sequence wouldn't work because this sequence's amino acids are usually hidden by this time. So you need a patch that basically forms as part of the protein fold on the outside because this protein is folded by this point. For the ER signal sequence, that protein wasn't folded at all. So a sequence of amino acids that were sequentially together would make sense. Here you need the fold. All right, so basically after these two enzymes do their job, and you don't need to remember the names of these enzymes. I wouldn't know them if I didn't have them written down here myself, but you just kind of need to know what they do. They basically are creating this mannose 6-phosphate at the end of the oligosaccharide that's on our lysosomal enzyme. And so here's just an image of that mannose sugar. Carbon 1, 2, 3, 4, 5, 6, mannose 6-phosphate. All right, so a few questions for you. All right, so the mannose 6-phosphate is going to be added to our lysosomal enzymes somewhere in the medial to trans Golgi. So then it's ready to be loaded into a vesicle whose destination is the endosome and then ultimately the lysosome. Endosomes often become the lysosome as more and more protons get put in here. It becomes more acidic. So endosome can actually become ultimately the lysosome. So my question for you, where does the M6P receptor need to bind tightly to the M6P lysosomal enzyme, and where does it need to bind weakly to it? Well, if you think back when we were talking about the retrieval, for ER retrieval, the receptor needed to bind tightly in the Golgi because that's where it's grabbing it, and then needed to release it in the ER where it's letting it off. So you always want the receptor to bind tightly where it's picking it up and release it, bind weakly where it's dropping it off. So the M6P receptor has to bind tightly in the trans Golgi and then weakly, thus dropping off the lysosomal enzyme when it gets to the endosome. So what do you think might mediate that tight binding in one place and weaker binding in another? Well, just like what we saw for the ER retrieval, it's a difference in pH. Only that difference in pH was only like 0.1. Here we have a much even bigger difference in pH as you get to the endosome. So that difference in pH changes the fold of the receptor and the lysosomal enzyme just enough that affinity is changed. And so we have high affinity here, goes in the vesicle, and when it gets fuses with this endosome where the pH is lower, that fold changes a little bit and this becomes lower affinity and the lysosomal enzyme's released. So you want high affinity where it binds, low affinity where it's released. Now, sometimes, even as good as this is, sometimes a lysosomal enzyme might accidentally get put into a vesicle that's by default headed for the plasma membrane. So how might they be able to be retrieved? Well, the key here is that some of the M6P receptors are accidentally leaked to the membrane as well. So there's a lot of empty M6P receptors on the outside of the plasma membrane, just waiting for some of those lysosomal enzymes to be released there, and then they grab them, and once they grab them, they will actually bring them through the endocytotic pathway to the endosome, so they can kind of retrieve those lost ones. So how do we retrieve the accidentally leaked ones, the lost M6P lysosomal enzymes? Well, we purposely leak some of the receptors out there as well to go retrieve them. This is kind of like that whole idea we talked about again with the ER retrieval of sending the lifeguards out where rescuing is going to be needed. So you send some empty receptors out where the rescuing is needed at the plasma membrane. Bring them back in to the endosome through the endocytotic pathway. What do you think happens when lysosomal trafficking is malfunctioning? Well, if lysosomal trafficking is malfunctioning, your lysosomal enzymes don't get to where they need to go, to the lysosome, and then you're going to have a problem with waste buildup. And that problem leads to diseases. So here's some lysosomal storage diseases. One of them is called Hunter syndrome. This is the inability to break down mucopolysaccharides. So one of those enzymes that is really important for breaking down mucopolysaccharides is mutated and therefore not functioning. So the lysosome's still working, and it can break down everything else, but that particular enzyme is missing. And so this leads to some characteristic symptoms and phenotypes with this. But for the most part, these people live kind of relatively long and happy lives. Not completely kind of as we would unfortunately call normal lives, because they are a little bit disabled, but for the most part they are fully functional, happy, healthy individuals. Right? This is another syndrome called Hurler syndrome. Right? Same idea. Both of these are defective in a single lysosomal enzyme. So in other words, the enzyme that does a particular job is mutated. However, we have an eye cell disease as well. This is much more problematic. Both of these diseases, the individuals live pretty long, relatively healthy lives. Not completely normal. They have some illnesses obviously associated with them, but still live long, relatively healthy lives. Someone with eye cell disease, though, this is a very dramatic phenotype. Unfortunately, this causes growth retardation, so they stop growing right around 1 to 2 years of age. Major respiratory issues, major motor control issues, and most of these patients unfortunately die certainly by age 4 or 5. Now, why is this? This is still a single gene that is mutated, just like these. These were single genes that were mutated, but these were single genes for lysosomal enzymes. So only that lysosomal enzyme was affected. This happens when the gene that's mutated is involved in getting the lysosomal enzyme to the lysosome. So in other words, the trafficking. And if you remember in another one of our asynchronous lectures, I talked briefly about the fact that we use temperature-sensitive mutants to study these, because if you have a mutation that causes a defect in the transportation, everything that needs to use that transportation system is affected. It's not just that one thing. Well, here, even though this is a single gene mutation, if that gene mutation stops all transport to the lysosome, now all your lysosomal enzymes are defective. Here it was just the one with the mutation that didn't work. Here, the mutation is involved in getting all those lysosomal enzymes to the lysosome, so now all those lysosomal enzymes aren't where they need to be. You're not degrading anything. Here, most things are degraded, one thing is not. Here, everything is not being degraded. So the buildup of this trash, of the waste in your cells, grows much, much more rapidly, and unfortunately, therefore, this has much stronger, worse phenotypes and earlier death. So what types of genes could we be talking about that cause this? Well, you can have a deficient receptor. So if the receptor for M6P is the mutated gene, you have nothing to recognize that M6 phosphate, and therefore nothing to grab them in the Golgi, cause them to be packaged in the correct vesicles and sent to the lysosome. If nothing can be sent to the lysosome, all the lysosomal function is lost. That's one gene that could be defective. Either of these two enzymes that are required to make the M6P, if either of those were defective, that also would cause all of our lysosomal enzymes to no longer get the M6P they need. Therefore, they wouldn't be recognized as lysosomal, they wouldn't be put in those vesicles, and they wouldn't be sent to the lysosome. So if any one of these three genes are the ones that are mutated, it's going to affect all of your lysosomal activity, thereby causing this much more dramatic disease, eye cell disease. All right, so this is mixed group problem number 7. I'm going to kind of walk you through it a little bit, but hopefully by this point in the unit, you've really started to think about how you can answer some of these things. So let's say you were a part of the important discovery that determined the importance of M6P groups that mediate sorting and delivery. How would you demonstrate this importance experimentally? So you think M6P is important to get things to the lysosome. How do you demonstrate that? Well, we've talked about the two aspects to that. If you think M6P is important, you basically cause M6P to be loaded on a reporter protein like GFP and see if that gets to the lysosome now. Second, you can take the M6P off of your lysosomal enzymes and see if they no longer get there. So if you remove M6P and they don't get to the lysosome, that's a pretty good indication. If you add M6P to something else and now it does get to the lysosome, that's a really good indication. Right? So you know that the sorting signal is only part of the story. What else is needed to help these proteins arrive? Well, this is all that other machinery we've been talking about. You need a receptor that binds to the M6P signal. You need all the vesicle sec proteins and all those other proteins we talked about that help kind of aggregate all the same cargo together in a vesicle. So you need that type of machinery. Then you need the coat proteins to cause the vesicle to bud, and you need the appropriate RAB and snares, V snares, to get it to the right spot. Right? So all that machinery we've been talking about is still in effect here. Now, the family members of the sec proteins might be different if you're going to the Golgi versus the ER versus the lysosome, but you still need those proteins. Right? So you need the receptor, you need all the machinery that loads the same cargo up into the single vesicle, and the machinery that buds that vesicle and fuses it with the lysosome. This one's a little bit harder. How about you try to identify these other proteins? Well, hopefully throughout our lab techniques series in lab, you'll kind of have some ideas. But there's several options. One is co-immunoprecipitation. So you can add your M6P protein, and you can basically allow it to bind to whatever it's going to bind to, and then you can lyse the cell in a way that doesn't cause protein-protein interactions to be lost. And then if you immunoprecipitate, if you use an antibody against your M6P protein and pull it down, anything it's bound to will come with it. And so we can kind of then identify all those things that our M6P protein was bound to. Right? You can isolate vesicles with the M6P proteins and see what the coat proteins are. There's a number of ways. Yeast 2 hybrid will look for binding interactions, things like that. So just think about this a little bit and do your best, but those are some ideas right there. In your studies, you realize the initial sorting of proteins with M6P is not perfect. Some of these proteins leak and accidentally become secreted by default. How might these leaked proteins still be able to make it to the lysosome? Well, I'm not going to answer this one for you. The answer is actually earlier in this video, but think about that and try to answer that together as well.